I'd like you to come up with a hypothesis to explain this observation.
100% on the mother and 78% on the father.
Well, the mother's eggs would have to carry the same chromosomes previously, so some kind of change of the meiotic division of the egg.
Sort of.
What is fraternal twins? 2 eggs.
2 eggs.
Okay? 2 eggs, 2 sperm, just held in the same development. What is identical twins?
An egg that splits in half.
Yeah, everything has happened, and then the cells split. So you end up with 2 identicals. They have the same genotype. Well, in this case, they have the same genotype from mom. So how many eggs did we use? 1. But 78% identical from the father's side. So how many sperm did we use? 2.
2 sperm.
This is interesting.
For 1 egg.
So should that have happened? No. So the only way this could have happened is if the egg thought it was fertilized, went through cell division, and then got fertilized.
Mm-hmm.
This is really rare. So we have some genetic information that says it happens. Things can happen spontaneously. All reactions are
spontaneous in either
and
direction.
if the cell thinks it has been fertilized, a calcium wave will just take place. Spontaneously, it will take place. The jellicoat will form, chromosomes will align, replicate, so forth. Well, at some point, if you are not fertilized, it should say, hey, there's not enough genetic information here, abort. But let's imagine we have extra initiator proteins. Now we cause replication to take place of the mom's genetics. So we've duplicated. And reaction takes place. What could we create? So we do not have fertilization, but we have duplicating of the chromosomes. So do we have the correct number of chromosomes? Yes.
Mm-hmm.
What have we created? It's not a clone. Okay, because we went through meiosis. So we have different allele frequencies, different allele combinations, so it's not a clone. But what did we create? Talk to Chet for a second. Try to come up with what we created.
What did we create?
That was funny.
What have you created? Say it. Jesus, there's a problem with this. It's immaculate conception.
Yes.
There is a problem. Could it be a male? No.
No.
It has to be female. This the only way it can work is if it's female because you got the male from mom, or from dad. You couldn't have got it from mom. So the X would be XX chromosome would be female. It would be different from the mom because it's a selfing. Even selfing, you still got meiosis taking place. But you would get immaculate conception. Now, it is fairly common in some animal kingdoms. So sharks can do it, certain rays can do it, fish pretty commonly. There's evidence of some amphibians to do it. In fact, in mammals, there are lineages that we know, hey, this female was never exposed to a male and all of a sudden has an offspring. In humans, there is evidence it might have happened one time recently. There is a little girl, she was in Brazil, and 13 or so year old girl gets pregnant. She is claiming did not have anything. What's the first thing you think of? Definitely dad or uncle did it, right? Mm-hmm. That is what we're thinking. The genetics, though, says it was a little girl that was born. Very, very close similarities to mom. So like 98% similar to mom in the allele frequency. So what does that tell you? What could that be indicative of?
That it was like developed. Like,
It could be immaculate, so you had this is selfing, or it could be in a case of extreme inbreeding. So over many generations of extreme inbreeding, your allele frequency become more similar to each other. You get homozygosity of alleles. So that could be. So there's two possibilities: extreme inbreeding, or this is a case in humans. Don't know.
Did they
Right.
see how much she's related to?
Yeah, they only looked at mom's genotype to daughters. So they did not look at families. So it'd be interesting. So you'd have to then look at dads. Is this multiple generations of inbreeding, or is this a case of selfing? So things do happen. Remember, all reactions are spontaneous. Shit can happen. Give enough time, enough individuals, my laundry should come out folded.
What if this were to happen with someone who's intersex? Could it be avoided?
Intersex is super complicated. Great question. We're going to get to sex and development next quarter, but so you are aware, biologically there are three sexes, okay? There are male, female, and intersex. Intersex is a continuation. Trans is not intersex, okay? Trans is a behavioral aspect, can be controlled by any number of different things, psychological, behavioral, hormonal, whatever it is. That is not intersex. Intersex means typically ambiguous genitalia. May require hormonal therapy, may not. Actually, in our country, when you hear doctors are performing hormonal therapy and gender reaffirming care on minors, no doctor on earth is changing a little boy to a little girl before they are mature. That's just not how we do this in this world. But you might hear that that little boy went through gender reaffirming care. What that might mean is that his penis was split, or there might have been urethra problem, or that the hormones did not engage and we do not have clear development. So you might have given a spike of hormones at younger, we might need to do something. So that would individual have been called intersex. So intersex can range from slightly ambiguous genitalia to true hermaphroditism. In our society, this is a controversial topic because people don't understand the biology. Intersex is also not rare. Intersex, the last data I saw on it was somewhere in the neighborhood of 1 in 60 or so. So that means we have 60 major students. Statistically, 1 would be intersex. And here's the other thing. A lot of people don't even know they are intersex because it got gender reaffirming care when they were young. So they were born with X, Y, or Z. That was fixed. No big deal. So this is an issue in development typically. So Y chromosome shuts off. There's an abnormality, a deformity, something like that. We'll hit that. Good question. All right, here's our first question. I'm trying to get on the spring quarter over. You got me going into spring. This is the fun stuff. We can actually start diving into animals.
It's like all the questions I've been asking.
Yeah.
Spring quarter.
That's spring quarter. That's next quarter. But we're getting there. We're so close.
Yay!
Feel it.
So close.
What does that mean for like Trump's new thing? Is like that there's like whatever like you're.
Yeah. I really am not a political person. I really am apolitical. But if you stop gender affirming care on young people, it's cruelty. So imagine you have that little boy who has a urethra problem. That is gender reaffirming care. So you don't fix that, that is a medical problem. You don't give the supplemental hormones, that is a medical problem. You're stopping normal development. So there's a lot of ignorance in this world, especially on this topic, because people. So if we were to start this conversation off with a layperson, okay, someone who doesn't have a science background, to teach them a concept of evolution, it took us 4 weeks to get there. To teach you guys about development, it took us 8 weeks to get there. So can I have a 5-minute conversation with somebody who doesn't have any idea about this and convince them? No, it takes a long time. So theoretically in our society, you divvy up your experts from your novice, right? You theoretically listen to the experts. Well, every once in a while, the novice start taking power back and things go badly, and then they'll reset again. Because, measles happen. A buddy of mine's doctor in Snohomish County, he messaged me. He's like, measles are in Snohomish County. I can confirm. And I was
Oh,
like,
great.
shit. He's like, saw one today. And I was like, fuck.
Keep Sylvia at home.
That's when he was basically like, we should keep Connor at home. All right, we're not doing too good on this question. Talk to Sharon.
I picked C, A, B.
I think I don't even know, but I think I need BAC.
But there was a similar question on the practice. Like, in what order do the following processes occur?
So, but as soon as the MMR is 12 to 15 months, 12 months is a little on the young side, 15 is So this is a good question. Are you vaccinated for MMR, measles? If you've had your MMR vaccine, you went to public school, chances are you're vaccinated for it. Not a big deal. The vaccine efficacy is around 99%. The problem is young people, like, under 12 months is when you can start getting the MMR vaccine. 15 months is like ideal timing. So if you're under 12 months, MMR is you don't have it. So Sylvie does not have measles antibodies yet. However, by breastfeeding, she theoretically is getting some from Charlotte. Okay, so hopefully she has immunity. Now here's the thing, herd immunity is when you vaccinate enough of the population that it will not spread. Okay, typically you need around 94 to 95% of your population, which is perfect because there are a subset of people who cannot get vaccinated. For whatever reason, they're allergic to eggs, they're allergic to something, they've had responses. So they have an autoimmune compromising where you can give them a trillion vaccines and it just won't uptake. So you herd immunity, you get about 95%, it's not going to spread anywhere. The problem is anti-vax is increasing. Now here's the deal. The vast majority of Americans are pro-vaccinations. The problem is it slipped to about 89%. Okay? So at 89%, you're getting a good portion of us are vaccinated, but you're not at herd immunity. You really need that 93, 95% threshold. So it is going to spread in communities. Now, it is much more likely to spread in communities with lower vaccination rates. The one in Texas and New Mexico have much lower vaccination rates, for whatever reasons. Okay. So with this, fertilization occurs. Sperm meets egg. Calcium wave takes place. Jelly-coated forms, and we get completion of meiosis II. Okay. The polar body gets ejected, and then we undergo
DNA replication.
and then we form cleavage. And we go cleavage, 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 cleavage to form our blastula. Okay?
Yeah.
So, I want to go from a ball, an egg, to a cute little puppy. Okay? How do you do that? What's the first step? Fertilization. Then we do DNA replication, cleavage, and then we do lots and lots of cleavage, right? I need to make a huge ball of cells. At this time, what are those cell types? What's the types of cells we have? We made a whole bunch of them. What do we call it? It's a blastula, but what type of cells? What are our types of cells again?
Ectoderm, mesoderm.
That's tissue layers. Types of cells were totipotent.
Totipotent.
It means it could be anything.
It could be
Pluri
anything.
means it could be
Lots.
many different things.
Many different things.
Determined means it can be
One.
a few things. Differentiated means it can be
One thing.
one thing. At this time, we have a ball of big cell, a blastula. What type of cells are we? Totipotent.
Totipotent.
Any one of those cells can be anything. Okay, so if you wanted the clay, you could do this. The clay's kind of heading on the wrong side of age. You start with a single cell, you cut it in half. Now you have two cells.
Two cells.
First cell division, now you have two cells. The cell size got cut in half. Then you go to 4 cells. The size of those cells got cut in
Half.
half again. Then you go to
8.
8 cells, 16, 32. Holy shit. Really quickly, you get a lot of cells. And so all of those cells can be any number of anything. They are all totipotent. So after the third division, how many cells do we have? Has the embryo gotten bigger than the zygote? How are they adhering to each other? And which cell stage must be skipped? Dr. Trey.
8 cells.
First cell division would be 2. Second cell division would be 4, so it would be 8 cells. Is the embryo bigger than the zygote? I don't think so. What do you guys think? Are cells adhering to one another?
Did you say 12 or not?
I said
Like at point number 4. Oh no,
This is another great
first
question.
question. Did you say
Embryo
12?
the same thing as a fetus? No. Embryos are eggs. They're little itty bitty eggs. They have not embedded anything. We don't have a fetal development yet. We don't have a placenta. Okay? So people confuse stages all the time. So when we talk, we'll get there in a second. I don't want to skip ahead. How many cells do you have at this point? 8. Has it changed size? No. The blastula is the same size, but we have number of cells have shrunk each individual cell in half compared to the previous division.
Oh,
Are they adhered to each other? Yes.
Yep.
Because if they are not adhered to each other, what happens? Falls apart, right? So let's imagine at this stage it gets cut in half. That blastula goes and separates out. What do you now get? 2 zygotes, which could lead to 2 fetuses, which are twins. Now let's imagine it fell apart to such an extent, but it's still stuck together at points. What would that lead to?
Conjoined?
Conjoined. As conjoined twins. So abnormalities at this point typically lead to abortion. Okay? This is where I was 10 seconds ahead in my head. Abortion in this class does not mean artificial abortion where you go get it removed. Okay? Abortion in this class just means something failed, the fetus got aborted. The embryo, miscarriage, anything that can happen. Now, you might say, what's the odds of that, okay? 2 healthy 20-year-olds are 20 to 30-year-olds are trying to have a child, okay? At any given month, there's 1 egg released. At any given month, there's approximately 20 to 30% likelihood at fertilization that it carries a successful term, okay? About 20%. That does not mean that fertilization did not happen. Does not mean that you were not pregnant. It just means the rest of the time, it ended. Something went wrong. Disconjoined of chromosomes, abnormalities, to not fetal the alignment's wrong, placental alignment's wrong. Any number of things can give rise to abortion. Okay? At 30 years old, cut that in half. 35, cut it in half again. So that starts explaining why when you are at 37, 38 years old, your chance of getting pregnant is about 5%. Okay? What? The likelihood of successful pregnancy. Okay? So peak terms for pregnancy is before 20. And then they start dropping precipitously after about 26. Before 20. Correct. Yeah. Yeah. So not telling you to do anything. No, gross, ew. Okay, no children. But we can start explaining what happens, okay? So this is happening really quickly. Which stage of the cell cycle must be skipped? Doctor, what must be skipped?
There's a cell phase where they like rest and like grow back, right? Growth phase. Whatever. Is that interphase?
Anybody got an idea? What stage of the cell cycle must be skipped? G1, excellent. So we must not be doing any transcription. No transcription, no translation. We are making nothing. But all of those proteins are there. Where did they come from? Mother. Excellent job. They came from mom. Mom's transcription actually controls all of your early embryonic development. Your genes were not responsible for early development. So now let's start applying this. You're a doctor. Your patient continuously has self-abortions. But they're alive. They're fine. They're normal. Where's the problem? Their transcription. They survived it because they were using their moms. So this is when you can have an issue and no longer be able to reproduce on your own, because even though you're alive because your mom is alive and was able to successfully do it. If you cannot successfully do it, well, that's a you problem. That is your genotype, not your mom's genotype, which controlled your development. So your development was controlled by your mom.
Your mom.
All right? So, ovary releases an egg. If fertilization happens, we get completion of meiosis 2. Get fertilization, we get the DNA replication, alignment of the chromosomes, and we go through cleavage. And we'll do cleavage until we create our blastula. At this point, it can embed or it won't. Okay? We'll do blastulization, then gastrulation, which will create your 3 tissue layers: ectoderm, endoderm, mesoderm. That's just a realignment of the cells. At that time, things will start realigning and turning things on and off. So if I'm going to be ectoderm, I'm going to turn on ectodermal tissue. If I'm going to be endoderm, I'm going to turn on
Endodermal tissue.
Excellent. Those are going to be receptors that tell it, hey, I am ectoderm. From the reading, please tell me what the receptors were and what are the signals for ectoderm.
Did the reading last night at 11, so I don't remember anything. Did you do any of the reading?
I did, but I don't remember anything about it.
Well, at least you took some notes. Everyone took notes.
All right, what is the signal, the receptors? Anybody remember? We need to make sure we're doing the reading, okay? Noggin and BMP4. All right? These will come back to haunt you a lot over the next little bit, okay? Noggin and BMP4. So cells will rearrange based on what types they are. So we're going to be ectoderm, endoderm, mesoderm. And because of that, different genes will turn on and off. We'll go from totipotent to pluripotent. All right? If I am mesoderm, I'm only going to make mesodermal stuff. Okay, I'm not determined to make anything, but that's what I can now make is mesodermal. If I'm ectoderm, I'm going to make ectodermal stuff. We're going to continue to rearrange and to make our full tissue layers. At this point, we're going to create, we're going to make a tube, okay? At the beginning of last quarter, we talked about you can be we're deuterosome development, means we make our mouth second, okay? So protosome versus deuterosome. All that means is the directionality of that tube, okay? You made one end versus the other. Tissues then get pulled through and we undergo gastrulation. At this point, we have 3 tissue layers. So, fertilization, cleavage, flagellation, gastrulation, and then we'll do neurulation in a bit. Let's see if this works. At this point, we have pulled through, and then we're going to invert, and you will see the cells kind of move right here. This is called Henson's note. We'll talk about that. And we form, it rolls over, and that's neurulation. So the tube gets formed first as gastrulation, and then we have neurulation. You'll notice that the cells are very dynamic. They're moving all the time. Things are moving and going around. Okay, so do it again. Okay, so from this video, talk show, what do you see? And then more importantly, what do you not see happen? Something is not happening. Alright, so what did we see? Saw cells moving, right? You formed your 3 tissue layers. You see cells folding. They're clearly touching each other. What did we not see? Should have been pretty obvious. Something did not happen. What? No, we saw them divide. We see them moving around. We see them doing things like that. Something did not happen.
Something that should have?
Nope.
It's not getting bigger.
No. Not getting bigger. Excellent. So we're not putting anything in. Did any of them die? Did we see apoptosis happening?
No.
No. That tube was not formed because you just killed things off in the center to make a tube. Tissues arranged and pulled through. So I have a question for you. Let's imagine a single cell right here dies. What happens? What? What'd you say? Say it confidently. Yeah. What happens? Single cell dies. A single insignificant cell dies. What happens? Abortion. Embryo dies.
Mm-hmm.
Okay? That single cell, because it is pluripotent, is going to make any number of different things. One cell might be responsible for a whole heart. One cell might be responsible for a whole limb at this point. So if I have a single cell death, what happens?
The whole thing gets aborted.
We abort everything. So at this point, we're still 2 weeks in. Things can start being very dynamic and changing. Everything has to go perfectly, otherwise we end it. So at a healthy person, young, you're only getting 20% of successful fertilizations even make it on to becoming an embryo. What happened? What went wrong? Cells died. A single cell might have died. A single chromosome misaligned. Any number of things, and we ended it. Okay?
So it's
Yeah,
more likely
absolutely.
to the thing to die instead of like basically.
Yeah, so you're going to put 9 months worth of energy and tens of millions of calories into something, it better work, right? So you might as well get rid of it if there's any issue. And that's what happens. Okay, in experiments, we're going to disable RNA polymerase, okay? So if I shut off RNA polymerase, what does not happen? Transcription does not happen. All right, Dr. Sher.
I put paternal genotype.
I put maternal,
That was good maternal.
maternal, because I think it's like he said with the mother doing the translation
Oh yeah.
to get the or transcription.
Okay.
So, do
Yeah.
I have RNA polymerase? No. But you're still doing what?
The
Transcribing and translating and making proteins, clearly because we have initiator proteins. Clearly we have other things like kinetochores and all the other stuff. However, what does that tell you? It's maternal genotype. Mom is doing everything.
It's just you are missing the RNA.
Yes, correct.
Yeah.
So, you're not doing anything. Your mom's genotype is doing everything. Mom's genes are expressing. So this is really kind of cool. So our first bit of development, right? Go from a single cell fertilization. We create a blastula, lots of different cells, so a cleavage to a blastula. This is the rearrangement and gastrulation. Okay? What type of cells do we have here?
site.
One type of cell, right?
One cell.
It's an egg. It is totipotent. It's going to make
Anything.
anything. This is going to make anything. This is going to make anything. This is going to make
Anything. Most
few
of
things.
Few things.
Right? Many things. So, sorry, not anything. Many things. But we have
Many.
gastrulation. We've rearranged. Okay? As we start developing, then we're going to go and make these stupid things. Right?
Yep.
So I've rearranged and now have our three tissue layers. I'm going to go through neurulation, organogenesis, and then we're going to go through growth. Okay? Throughout the pregnancy and throughout development, truthfully, apoptosis just doesn't happen. Apoptosis doesn't happen until the very end. So here, actually in lab this week, we're going to pull out the human fetus collection. And you're going to look at human fetuses. And you will notice that some of them have no holes. They have webbed fingers. Why? Cells have not
Developed.
died.
Oh, no.
Cells
Excellent.
didn't die.
Let's also then start applying this to other tissues like organs and brains. Will you have all the ridges in your brain? No, because that you need cells to die. You need to create more surface area, you need things to die, right?
Mm-hmm.
We haven't done that yet. So what does that tell you about the brain? It's not developed.
Mm-hmm.
Right? We haven't developed. Well, your guys' brains might be just coming up on completion of development. So, Dr. Chair, recap. What are each of those?
Toti means anything. Pluri means most of everything. Determine means few things, and differentiate means one thing.
Toti means,
Anything.
pluri means,
Many.
Most of anything.
determine means,
Few things.
differentiated means.
One.
Unique.
Unique.
There we go. Totipotent, is during egg through blastula. Pleury is blastula through gastrulation. Determine, we haven't got there yet. All right, that's next quarter. These are next quarter. So our goal is to get through pluripotent this week. Okay? This is chemistry. Sorry guys. My bad. I talked to Sure.
I picked Toady. I think I picked Toady the differentiated.
I said the opposite, where it's differentiated to toady.
I did. Yeah, mine is not good. I did pleury to toady. What did you did the last one? It's the same one you did.
Yeah. Because skin cells would be differentiated, and then they would need to go to toady to create stem cells to then create anything.
What is skin cells?
Differentiated.
Skin cells are skin cells. Therefore, they are
Differentiated.
differentiated. And in order to make a whole new organism, I need to be able to go back to
Toady.
toady. So 4 is the correct answer.
You're right.
All right, I have a question for you. Can cloning, based on just the knowledge you have today, can you say, I want to do me? All right, the world just needs that. Can we make a new Ricky? Why is that wrong? Cloning cannot ever happen, nor ever will. Talk to each other and try to figure out why.
Because you're basically using the same genetic material. And if your clone gets sick, you get sick. That's what we learned last quarter.
Oh, like the bananas?
Yeah, like the bananas and the oranges and all the fruit.
Yeah, you
If
told
you
me.
clone it and that clone gets sick, you get sick.
Yeah, but
Because the
You
clone
could
is
still
basically
make it, no?
the clone is basically the same as you. It's the same genetic.
so they can't survive in the same space, I would think. So
And
All right,
I don't think
somebody want to tell me why cloning can never truly actually happen? No?
You can't get the same chromosomes.
Every single cell in your body, and you can't determine whether
No?
Is it because the formation of like when it becomes totipotent to differentiated, it can happen to different cells at different times.
Okay, keep going down that path.
It can be endoderm or it
No.
The
You're
other path.
copying my genetics.
Yeah.
What's wrong with that? You need my mom.
You need the mother.
Because who controlled my development early on in utero?
Your
My
mother.
mom's genetics. So now, okay, you say, okay, cool. You're going to take this elderly woman, right, and we're going to embed a new fetus embryo into her that's a copy of me. Okay? So theoretically, that's as close as you could get because then it would develop using her genetics. The problem is what has happened over that 37 years. She got older. Time happened. Changes happened. Is it the same environment?
No, absolutely.
Everything has changed. So, will you ever truly produce a clone?
No.
Is the closest you can get like right after the baby is born?
Yeah, that would be as close as you possibly could get. Even then, there being a fetus there, has that changed mom's expression, changed mom's hormone levels, changed everything? Yes.
Yes, everything changed because
Everything changed. So can we ever truly do this?
No.
No. All right. So let's say you really, really love that dog and you want to make a new dog, right? The same dog. Will it look similar?
Yes.
Yes. Will it look identical? No. It'll be slightly different. Will it have the same developmental pattern, same hormonal levels, same neural relation, all those things?
No.
No, it'll all be different. So this becomes a problem. Now, where cloning will take us is, let's say you need a new heart, right? You ate at McDonald's for 46 years straight, and you need a new heart. So what I can do is clone you a new heart, grow it in a petri dish, 3D print a heart, take totipotent cells, 3D print them by telling them what to turn on and off, and reassemble it. Build it in a petri dish that has a clone heart that is your heart. Now I put it in your body, life is good. We can head in that direction. In fact, there's a really good TED Talk where while he is doing the talk, a kidney is thrown on the stage. So that's really cool. So we're headed in this direction, but truly, true cloning of an individual will never happen. Now, you could go down the path and say, well, I've created the perfect individual, now I want millions of them. And make that.
Eugenics?
Right, yeah. Eugenics could very easily. Just don't tell anybody in the bad science jokes. Okay, 10 seconds.
Ooh,
All right, talk, sure.
I wanted to
I've
change it.
experimented the sperm because that probably looked like the best answer based on what was given above. What else? Yeah, because zygotes are basically like toady. They could be anything. And sperm plus
All right, let's go through each of these.
zygote is bad.
What type of cells are these?
Toady.
Totipotent, excellent. That is
Determined.
determined or differentiated, depending on what type of muscle cell, right? This is a,
Differentiated.
that's differentiated, that is
Determined.
toady.
Toady.
Because eggs make
Anything.
anything.
Eggs
That
make
is anything.
Mm.
This is determined
This
to
is
make
differentiated.
sperm. A is the correct answer. Next question.
Mesoderm cells.
Dr. Sheriff.
Wait, what?
I picked Pluri. You got Pluri?
Yeah, I figured either Flurry or Determined, but
Yeah, I was figuring like Pluri or Determined, but I picked Pluri.
I went with Determined, but
Determined potent.
Dude, I don't have it though. Oh my god, she's like, I bring my exam back because we graded it, but she put my grade book. She said she wants it back. I think if we just saw I thought we if you wanted it, you bring it back. No, she said I wouldn't, but bring it today.
What can mesoderm mean?
middle.
lots of things.
Many different things, lots of different things.
Yeah.
So it is
Pluri.
pluripotent.
There we go. Mesoderm. Mesoderm is basically the middle.
And what's in the middle? A lot.
And it gives.
A lot of things.
All right,
Anything.
go to the
more
next
things in the
session.
middle.
Trying.
Oh.
Okay, 10 seconds. All right, Dr. Sheriff.
I picked the top three. The mother influences blushulation, gastrulation, and tells the embryo how to go to the
I picked B. And what else? And then E. That's what I picked.
Yeah, I put mom's action on A, B, C, and E.
Not all three of the moms.
Does mom control blastulation?
No.
No.
Yes.
Yes.
Oh.
That's what mom's genotype doing. Mom's producing all of those initiator proteins, RNA polymerases, all of those things to do it. Mom's producing all the nucleotides. She produced all of the DNA polymerases, 3, 1s, ligase, etc. She produced all of that. Mom's genotype influences gastrulation, but not truly. It's really blastulation. Zygote genotype takes over and tells it how to go from toti to pluri, which cells do that. And then transcription receptors are what changes. So a receptor says, hey,
I got it well.
I'm ectoderm. Hey, I'm endoderm. That's what's really setting it up. So 5 is the next big one.
A, C, and E? I mean, one.
ADE.
ADNE.
Zygotes?
Yeah,
No.
zygote's genotype tells the embryo how to go from toti to pluri.
Oh, I thought it was mother. No, zygotes?
You have 3 tissue layers, ectoderm, endoderm, mesoderm. Your ectoderm is probably what you're most familiar with because that's what makes up your skin. However, ectoderm is not initially meant for skin. Okay? The first animals did not have skin. So ectoderm is a byproduct. Our skin's a byproduct from ectoderm. Ectoderm initially is neural tissue, it's receptors, it's how you interact with the world, right? Your senses, how many senses they have now, dozens, right? Those are how you interact with the world. That is ectoderm. Endoderm is your internal interactions with the world, so your gut systems. Mesoderm is all of your muscles and hearts and organs and tissues. And so that's what we're going to spend a lot of time next quarter. So we're going to focus on endoderm and ectoderm this next couple of weeks, and then go move into muscles. So, sperm gets released, meets egg. Fertilization takes place. Meiosis II happens. Calcium wave takes place. Jellicoat expands. We undergo the first cleavage, then second cleavage, third cleavage, fourth cleavage, so forth, and we've created blastula. All of those are totipotent. We then undergo a rearrangement and the production of receptor proteins, what tells it to go from toti to pleury. That is gastrulation. Neurulation is what we're going to do next. And that basically says this is where we're going to set up our neural tissue. We're going to orient up and down, and so that we can create what's called a primitive streak. So if we were to set an egg against the wall, okay, and we're going to start it at one spot and run it down, that's what's going to happen. And that running down is called neurulation, and that's going to tell it how to build it. And that's your primitive streak, okay? So between frogs and chickens and goats and very similar, okay? All of that was set in motion by these cells changing, and it's a signal pathway. So this is called Henson's node, is what actually sets it up. And Henson's node just tells it, this is how we're going to build a body plan. I'm going to orient in this direction, we're going to build it. So, like any good scientist, you say, okay, let's screw with it. So I'm going to take a Henson's node from one animal and then plant it on an egg of another animal. Take the quail, stick it right next to the chicken embryo, and I get a
Quail.
quail with the genotype of a chicken. Interesting. Why? Doctor, what?
What's a quail?
Quail?
Yeah.
It's just a kind of bird.
Oh, okay.
So what does this tell you about the development of this animal?
It would be
A couple of cells tells the entire animal how to be built. Do you and a quail have the same genes responsible for ectoderm, endoderm, mesoderm? Yes, of course you do. Do you have the same genes responsible for building a heart?
Yep.
Same thing for a muscle?
Yep.
Yeah. So all I need is a handful of cells, but we're going to use slightly this different variant. So that's really kind of cool. And so today, our goal here is to understand neurulation. So the good news is we have a worksheet.
Yeah.
It's only one page.
That's good
It's
now.
the hardest worksheet you'll ever experience in your entire
Is it like
life.
this?
Yeah, oh
Boy,
God!
when you said one page,
Alright.
it's like, oh, it's
So-
one page.
Yeah, you're like one page.
It's one.
This is called Minions in a Coffee Shop. And the reason why is neurulation is crazy. If you've ever seen a minion, they're crazy. And so we're going to take something that seems to feel really simple, make it really complicated, which it really is, in fact, really
It's
simple.
one page back and front.
So
Oh, well, it is one.
if I have something that shuts something off, we call that an inhibitor, right? If I shut something off, that's inhibiting something. If I shut the inhibitor to the inhibitor off, that is an activator. What if I have an inhibitor of the inhibitor of the inhibitor? Now we've
Back.
It would shut
it
things
would
off.
shut it off again. Ooh, that's interesting.
Sort of like negative
Okay.
times negative
So,
times negative. It's still
let's get
negative.
Mm.
Yes.
Yep. We still have the doctor.
Dr. Dooley rocks.
I didn't like that.
We did.
Yeah.
Oh,
No, that's
We wrote it.
You wrote it.
Well,
I heard
Looks like you wrote
it's
it.
not.
Good job.
It's one page.
I want to show him I don't have that bad
Interesting.
writing.
Like all worksheets, it's gonna take the rest of the class. Okay.
Noggin.
Two signals, noggin and BMP4 work together during neurulation and determine which parts of the ectoderm become skin and which become neural. Use experiments 1 through 4 to answer the following. What is the default state of the ectoderm?
Let's see. This one is
Top is ectoderm, middle is mesoderm, bottom is endoderm. What is the default state of the ectoderm?
normal.
Normal-ish. Determine which signal prevents the neural tube formation and explain answers.
It is annoying.
I'm so confused.
Not just like
You are shown the three.
Yeah.
Does it say week 11?
Wait, what would you say is? Are they talking that this is? Wait, this is the left, right? So ecto, meso, and endo.
Yeah.
Is this what they're talking about? At the end, endo? I mean, ecto, meso, and endo?
I'm not.
Let me ask.
Because finals week has different starting weeks. So finals this week starts on the Tuesday.
You can look at, it's all your courses.
Okay, so yeah, that's what it is. So you said the first one would be
Yeah,
normal, WT.
yeah, I think so.
Determine which signal prevents narrow tube formation and explain your answer.
What signal prevents it?
Hence the syllabus gets long.
Signaling molecules have been altered.
But they wanted it to be it's not just finals are technically supposed to be
So they wanted it to be like way down at like
I don't get it. Which are the
I don't
ones?
get the question either.
The signal, do you understand?
Do you understand the question?
1B or even 1A?
Yeah,
1A we put normal, so we
1B.
I saw EMP4 because
In which one, I'm sorry?
EMP4.
The number
In
one.
1B?
Yeah.
In 1B.
Because in experiment 3, it's just EMP4 and the neural tube doesn't form.
Oh, there we go. Yeah.
In experiment 3?
Yes.
Or experiment 2? 3 is,
No,
What
no.
does WT
there's no BMP there, right?
Normal.
normal? Excellent. Means wild type. All right. First question.
Normal wild type. Oh. I know that one. I know that one.
Maybe I could be wrong.
One word chick for 2 or 3 students. Median. Okay, so.
All right. Few more seconds.
So it's not an inhibitor.
Ah, man. I should have killed it 10 seconds ago. Because we were at 100%.
And somebody changed it?
Somebody changed it. I don't want to throw them under the bus, but it is B. We're not going to, you know what? Throw 'em under the bus.
Okay, so I'm sorry, you were explaining.
So in experiment 4,
In
or
experiment-
sorry, experiment 3.
In experiment 3.
All right, next question.
Oh. I want to say that one. Oh, wait, can I change that?
Oh, the first stage?
First and older are the same thing.
Oh, jeez.
Oh, that makes sense. Why? Yeah.
English is stupid.
I'm not sure.
Not all cultures do this. Okay? So, garbage. I'm really good at picking out garbage ones. Hey, there we go. All right. All right. Development is weird. Okay? We actually measure when a fetus was their gestation from before they were fertilized. Okay? This is mom's last menstruation, is day 0. Okay? Then it goes to fertilization, whenever that happens, copulation. Then we go to day 0 is birth. So this is older in past. So the first stage of development is this, which is the older stage. Okay?
It was around for the longest amount of time.
And then, so for your bearings, this is about 39 weeks in human. This is about 38 weeks. 37 to 38 weeks. Okay. All right, we've oriented ourselves. Now we can work with this.
Oh, wait, you're talking about this right now.
No one said English was easy.
Using the data from experiments 5 through 7, determine where the signal comes from
Somebody
to
want to
machine
correct
error.
me if I'm wrong, but Southeast Asian cultures mark this as 0. So when you're born, you are 1. Is that correct?
Yeah.
Okay, I don't want to be wrong, but like,
It
I
might
think
be.
that's correct.
It might be. I guess it just depends on the culture.
But they call it 1. Somebody's culture know that for sure? That is correct. What culture is it?
Chinese.
Chinese? Okay.
Interesting. Okay, now can you tell me? Question, next question.
BMP4, because in experiment 3,
In experiment 3?
I thought
BMP4 that exists.
Which actually
Because there's no noggin. Noggin has been inhibited, which means BMP4
Oh.
is the only one.
Oh, because
Oh,
but
the
it says
noggin.
add inhibitor of the signal molecule noggin.
Yeah, so they've inhibited noggin, which means it's just BMP4.
That's very confusing for me. So then there's only BMP4. That's what you said, right?
So I believe that causes the neural tube to form.
Using the data from 5 to 7, where the signal comes from to machine. Five to 7.
mesoderm, ectoderm, endoderm, little sides. Using the data from experiments 5 through 7, determine where the signal comes from to initiate neurulation.
There. Wait, what?
That's what. Yeah, I put mine, but it's
I understand why you said this was the hardest one.
Yes!
Which signal most likely released by the cells?
You chose,
chose
an
A.
How do you know?
Normal routine. Huh? I don't understand.
Noggin. I think it. No, it can't be noggin.
Alright? Our first question. Our first actual like real question of the. Yeah, that too. It's only Monday. It's Friday. Can we just say it's Friday?
This machine is hard.
Alright? Talk to each other.
I picked neural.
Yeah.
Oh, I mean, that makes sense. Like, go outside. I know.
Well, looks like the nodal core, we can find it on the ecto, right? Ecto?
Yeah.
Oh, or ecto and meso.
When you think of ectoderm, what do you think of?
Skin.
Skin. But that is not the default state of ectoderm. The default state is
Neural.
neural tissue.
Neural tissues. That's right.
So, what did he say? Neural is.
Neural tissues
Neural is.
is the default ectoderm.
Is neural tissues is ectoderm.
Yep. Are you even paying attention?
I'm just trying to pay attention. I'm just asleep.
Sleep deprived? I am too. I'm really trying to get.
I am
really
sleep-
trying to get into this.
I'm sleepwalking.
You're sleep deprived.
I'm sleepwalking. No, actually sleep. No, you know what happens to my brain too?
What?
I hear something and I put it backwards. That's my problem. So then I'm like, oh. If I don't get it like really quick, it just, and then I'm like, wait, was it that way or that way?
Which signal is most likely released by the cells you chose as A? Noggin. Looks like.
Oh, group 2. Okay, group 1, meso and wow, what is the AP axis? Do you know what's the AP axis? How do you say X?
What?
The AP axis?
I don't know.
I don't know what that means.
I think it was just like.
Wait, where was it? Did I invent that?
I think no, yeah, that's in there.
Oh, it's up there. Number 2. Okay.
What's the default?
It's like just the center versus the side pairs is what I interpret it as. So like this center is the AP, and these are the left and right. That's what I thought it was.
Okay.
Well, anterior, posterior, which would be the
Oh, that makes sense. Anterior, posterior.
Which of the following schemes best represents the interaction between signaling molecules? Noggin activates BMP4, noggin inhibits BMP4, BMP4 activates noggin, BMP4 inhibits noggin.
Mm. What did you put in 3? I put is the last phase, but I don't know. What did you put?
I put each reacts to different signals in a different way,
That makes sense. But
which sort of matches the question I asked. Propose a hypothesis for why the endoderm does not respond to signals in the same manner as the ectoderm. I mean, they might react to the same signals, but in a different way. Or they'll react to different signals in a different way. Or the like. Oh.
is that what you have for 3?
for what?
Number 3?
I didn't come up with an answer for 3.
Okay. I put is the last phase, or and then I'm gonna add, or they react in a different way. I don't know.
Yeah. That's what I was basically trying to ask, like, propose a hypothesis for why endoderm does not respond in the same way to signals as the ectoderm.
In what layer can you find the notochord?
In what layer could you find
Would that be
endoderm?
mess?
Alright, next question for you guys.
Where does the signal?
Endo and meso? Ecto and
Where does the
meso.
signal come from? Okay.
Oh. Wait, what?
I'm just about conked out. I was doing all my homework yesterday, and somehow I did it till 11 o'clock. And I even studied for the quiz, for the OChem quiz.
Alright,
How many days you spent?
2 more seconds.
Oh, 5 to 7.
Alright, we're not doing so good on this question, so talk to each other, try to figure it out.
I just figure out 5 to 7. You said ecto?
Yeah, I said ectoderm.
I think, yeah. I think it's meso, but I did ecto too. And then I
Yeah.
I forgot to read 5 to 7. I went with group 1, so I was like,
Very good. Boy, this classroom is crammed.
All right, where does it come from? And how do we know? Well, let's do some experiments. If I cut out, so I have my wild type produces a normal looking development. If I cut out both the mesoderm and endoderm, I get
No neural tube.
nothing, no neural tube. So this must mean something. What does it mean?
Comes from either the end.
Yeah, it comes from something other than ectoderm. So the signal from ectoderm does not come from ectoderm.
Mm-hmm.
Ooh, that's interesting. Okay, but if I keep mesoderm in there, I get
Neural tube.
neural tissue. Therefore, the signal must come from mesoderm.
Mesoderm.
So then the question becomes, what is the signals? Close up the last one. One person still doesn't believe me.
Which signal prevents neural tube formation?
Seconds. All right, talk to your turn.
I picked BMP4.
I don't remember what I picked.
I picked noggin.
I think I picked Naugen. I don't remember why.
You're trying to
I need
keep
to
awake.
get some sugar though. Yeah. I don't do this.
Which one inhibits neurulation? BMP4. So BMP4 inhibits neurulation. There is a receptor on ectoderm. TGFB beta 1,4 something other, whatever it is. TGFB. That is the thing that receives the signal. Okay, I had this on an exam earlier in the quarter. The protein that you assembled in the first exam was you assembled a RNA molecule, and then you assembled the protein that will actually be receiving the signal from BMP4. So BMP4 inhibits neurulation tissue. So then the question becomes, how do you get neural tissue? Closing up the last one. Going.
Oh, okay. That's weird.
Okay, 2 more seconds. All right, so after turn.
I said that not the noggin inhibits BMP4 because in order to make neural tissue, and if BMP4 blocks neural tissue, you have to block BMP4.
So I put BMP4 inhibits noggin, but I don't know. Because he said BMP4 inhibits neurulation. No?
Yeah, if BMP4 inhibits neurulation and you want neurulation,
So
you have to block it.
BMP4 inhibits neurulation, which makes skin, which is cool. We like skin, but we also really want neural tissue, right? I want that brain. So we're gonna need to inhibit BMP4. So noggin inhibits BMP4, which then allows you to make your noggin. Noggin makes your noggin.
Noggin makes noggin.
Okay? So noggin inhibits BMP4.
Yes.
Just the dorsal fin. That's what we did last quarter. Last receiving hydrostatic skeleton.
Describe the role of the notochord during this phase of development in a few words.
Two. So it is a tube, right? You are a tube within tubes. It's from last quarter. Seal. How far first? We are 3%. It's a tube.
Ectoderm.
That's the first. Actually, I think of like the most comfortable. Spirit is the word. No one wants to go on Spirit. Their motto should be, we will get you there. Didn't say when or how good.
Oh, did you put find the notochord? Or you? Where do we find it? Then
Ectoderm.
notochord. Where do we find it?
It's in ectoderm.
Ecto.
How do you know that?
I put ecto and meso, but only in there. Describe the role of a notochord.
Ricky said it's just a tube,
It's just a tube.
apparently.
Yeah, it's just a tube.
So just to produce a tube, would that
No,
be?
it's just a tube.
It's just a
It's
tube.
there for aesthetic. It's like it's a nice tube. Change up
If
your
you have a
straw.
tube for your body, can you do something with it?
Become a straw.
Become a straw.
We're just really fancy
Thank you, we did do notochords last quarter.
I wasn't here last week.
That's not mine. They don't remember anyway, so it doesn't matter.
Yeah, it doesn't
When
matter.
did you do the first quarter vibe?
Last quarter.
Yeah, no, it's all on my mind.
Alright, with that said, we're gonna talk for a
Fall
moment.
2023.
About that.
About us not remembering?
Yes, exactly. That's actually coming up.
Oh.
So,
What's coming up?
TGFB beta 1 blah 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 protein. That is a receptor. That is on ectoderm. That receptor is not on endoderm, not on mesoderm.
Non-mesoderm.
So when you have a signal, is not going to be received by ectoderm and mesoderm, even though mesoderm is producing it. Noggin inhibits BMP4. It binds to the active site, preventing BMP4 from being able to bind. Okay, so noggin inhibits the inhibitor, which allows it to move forward. So why does this matter? Well, you want to be able to produce neural tissue, correct?
Yep.
Yep, that matters. What else does it matter? Why take this class? It's a real question for you guys. Dr. Chair, why take this class? What are we doing?
Learn.
Why do you need to know what a notochord
Bye.
is?
It's just a prereq for computer.
I don't even know why I'm here.
What class is this?
Like what?
Wait.
It's like the same question as why
What's
are we
the name?
taking calculus?
Like, what's the name of this class? Did
Why
you
are
say
we
that?
even taking calculus?
To learn evolution and
To learn
produce,
how the organism is formed.
interact, the
Right.
cycles.
Why should you
Sugar.
remember all this? How many of you again want to be a doctor, pharmacist, dentist? What do you think the very first question on the MCAT is?
What's
Neuralation.
another? That's neuralation.
This is this year's MCAT. All right. TGFB. Oh, shoot, it sounds familiar, right? Same exact questions. Okay? So if we do this expression and we change things up, how does this affect? Right? Now, you might say, I don't know these words yet. We're getting there, but you're starting to figure out, oh, what does this have to do with it? Do I have noggin? Do I have BD4? How does this work? And then we start getting this over here. What is a stem cell? How do they work? Right? What type of tissues? We haven't gotten to multipotent yet. Right? But you know what totipotent is. You know what pluripotent. Well, if I were to say, what's a notochord? From last quarter, what's a notochord?
It was
It's a tube.
It's
What
just
was
a
it
tube.
used for? What was its original purpose? To swim. Anterior, posterior, that's your tail. Okay? We co-opted that because we don't no longer use it any longer. We are not a coelomate. What was a coelom? Okay, what is it? Talk to her for 3 seconds. What is a coelom? Recap last quarter.
Trying to think.
No top bottom, just front back.
We're not high enough to do that.
Fun back, yeah.
Wait, I'm 18. I'm not old enough for that.
What's a coelom? It's the hydrostatic skeleton.
It's a hydrostatic skeleton.
It's
okay.
the tube to make sure well, we don't use that anymore because you have bones. What were the original bones for? Energy storage. Excellent. Do you think that's going to come back on your MCAT? 100 per 1,000 billion%. Your very first question. Question 2 is this. Question 1 is about noggin and BMP4. So if you do not know about noggin and BMP4, you don't know about this, can you be a doctor? No, you just got rid of your first 2 MCAT questions. About 100 usually.
Can we?
How many
Okay?
others?
You need to get probably about 95 of them right for you to get into medical school. Okay?
Yeah.
So What does that tell you about remembering stuff? Yeah, it's not going to just come back to haunt you next week or the week after. But do you think it's going to come back to haunt you in 6 months? Do you think it's going to come back to haunt you in 3 years?
Yes.
What about 5 years? 10 years?
Yes.
Yeah. When you become a doctor, do you have to go get do you have to do an exam every couple of years? Yes. And do you think if you forgot what TGFB protein does and noggin does, do you think they're going to allow you to be a doctor any longer?
No.
No. Because now you can't do things for your practice, right? We need to be able to understand it. Okay, so you made a kind of a funny joke, and then I'm like, but I got just, why does this actually matter, right? Well, this is why it matters. Because if you don't know this now, you're screwed. Okay? So we need to be up on top of stuff, actually doing it, working through every single study question, doing this, because guess what comes about? This. All right, so you say, well, I'm an environmental scientist. That don't matter. Cool. Tell me, how do toxins affect frog embryos and why? Is that important if we just dump shit in a pond? Oh, does it start affecting your TGFB beta proteins and affecting neurulation? Okay, now tell me, why are frogs more susceptible than a human? Our skin, excellent.
Sorry.
Thicker, waxy cuticle. Frogs don't have a waxy cuticle skin. All right? So now we just nailed environmental science. Okay, why they need to have it. So all of these things start coming back to each other. So you need a solid foundation, which we're getting now. All right? Thank you for opening that one up.
Yippee!
What I do?
Okay? I will see you guys either in labs or in class on Wednesday.